Greetings, students, and welcome back to part 2 of our unit on the tour of the cell. When we last finished up, we had just started talking about the different functional groups, and we talked about the first functional group, which is those organelles that are responsible for controlling and transmitting the genetic information, the DNA, the RNA, the ribosomes, that kind of stuff. And so today I want to start looking at some of the other different functional groups to finish up the chapter. The next group we're going to look at is the endomembrane system. The endomembrane system consists of multiple different organelles that do all kinds of jobs, including synthesizing material, distributing material, storing material, and also the exporting of materials out of the cell. First organelle I want to talk about is the endoplasmic reticulum, which is a network of tubes and sacs that are generally located in the vicinity of the nucleus, which of course makes sense because in the endoplasmic reticulum, especially if we're talking about the rough ER, you will find the ribosomes, which of course what makes it rough, which is where the proteins are synthesized during a process called protein synthesis. The smooth ER, which is similar in terms of a network of tubes and pathways and passageways and sacs, is a little bit different because it doesn't have the ribosomes, hence the smooth texture, and it's responsible for doing things like synthesizing lipids and processing things like toxins. The endoplasmic reticulum is an outstanding example of what we call a biosynthetic workshop because it's responsible for making so many of the different types of macromolecules we looked at in the previous unit, including lipids and proteins, which you can see right here. Here's an example of a smooth ER and rough ER, which you can see is in the general vicinity of the nucleus. The rough ER closer, and then the smooth ER on the outside here. The rough ER in the vicinity of the nucleus is very important so that the mRNA can carry the message to the ribosomes. Here, these brown dots here, you can see the ribosomes which line the passageways. Now what will happen is the messenger RNA will carry the message from the DNA to the ribosome, and in the ribosome, protein synthesis will take place. That's this growing polypeptide here. And then the protein may get picked up by some type of sugar or something, and then it will be surrounded by a vesicle, which will then pinch off from the endoplasmic reticulum and then travel on to the next stage if it's processing and packaging in the Golgi bodies or the Golgi complex. Okay? So in the smooth ER, the lipids are made, of course, remember, but in the rough ER is where we'll find protein synthesis taking place. The Golgi apparatus consists of stacks of sacs, which take the products of the endoplasmic reticulum and process them and then package them to be sent off to other organelles or to the cell surface for expulsion out of the cell. So if you think of this, the analogy of the cell city which we've been using, the endoplasmic reticulum would be like the industrial park where the factories are making things. And then they have to package those things and then deliver them, in this case they deliver them out in boxes, but in vesicles, to the Golgi apparatus, which you can think of as like the Postal Service or FedEx or UPS, and that, which then has the capability to repackage the material again and then deliver it all over the place. This is what the Golgi bodies look like. You can see another example of a bunch of passageways and pathways, and then you can see it also creating these little tiny vesicles with the material that it's going to be packaging up and then transporting out of the cell or to some other part of the cell, to another organelle. But you can also see the transport vesicle delivering the materials from the endoplasmic reticulum. Perhaps maybe it's a protein or something that's going to be processed up and then shipped away. Another organelle we're going to talk about here in terms of thinking about the cell city is the lysosomes. And the lysosomes are like the waste management company or the sanitation department. Lysosomes contain enzymes that are responsible for doing things like breaking down material that's been ingested through like phagocytosis or something, engulfing. It also can help break down things like damaged organelles and recycle that material. So it looks kind of like this. Imagine here a food particle being engulfed by this cell. A little vacuole forms around it, then the lysosome will come in, secrete some of those different digestive enzymes, and then break down those food particles so they can be absorbed into the cell. Functions similarly in the case of, say, you have a damaged organelle, like in the case of this bad mitochondrion here. The lysosome is going to secrete an enzyme to break down the mitochondria and then release those particles back into the cell. Vacuoles are larger vesicles that have a variety of functions. Some, including like for protists, they have what are called contractile vacuoles, which regulate the amount of water in a cell. You might also find things like small food vacuoles, like in the previous slide we looked at, or in the case of a plant, you might find a large central vacuole that's quite oftentimes filled with fluid, like water, of course. The food vacuole will carry food particles that perhaps the cell has ingested in through engulfing, in the case of an animal cell. Here's an example of a contractile vacuole here. Central vacuole here. And then again, just to review the movement of material again, from let's say the rough ER, the protein has been assembled, and then it will be shipped off via transport vesicle to the Golgi bodies, and then within the Golgi bodies, those particles will be then again repackaged, put into some type of vesicle. Perhaps what was produced here was some type of enzyme, which is then put into a vesicle. Then that could be something that might be used somewhere else in the cell to help facilitate some type of process reaction or something like that. Might be something that's needed beyond the cell. Maybe it's produced, I don't know, something insulin or something that needs to be then distributed out, say, for example, to your bloodstream. The third functional group we have your energy converting organelles. These include mitochondria, which are the organelles that carry out cellular respiration, which is the energy transformation that occurs in which the organelle takes the energy from sugar and converts it into a form of currency that the cell can use, which is called ATP. Mitochondria is interesting because it has two membranes. It's a double membraned organelle with an outer membrane and an inner membrane. It also has what's called a mitochondrial matrix, which is the inner part of the mitochondria. And inside it, you find things like DNA and ribosomes, and also some of the enzymes that are responsible for the reaction that takes place during cellular respiration. Here's what a mitochondria looks like. You can see the two membranes, the outer one and the inner one. You can also see these folds here, the cristae, which increase the surface area of the inner membrane because you have those folds. Chloroplasts are another energy converting organelle. In this case, they take the sun's light and convert it into a form of energy that the cell can use, which is sugar. It's another example of an energy transformation, like I said, and we call that process photosynthesis. Both mitochondria and chloroplasts, scientists believe, evolved through a process called endosymbiosis, part of what's called the endosymbiont theory, which states that scientists hypothesized a long time ago that mitochondria may have been small prokaryotic-like structures that were ingested by early eukaryotic cells. And those eukaryotic cells liked the mitochondria. They liked the chloroplast that they ingested, and those organelles stayed behind, and they developed a symbiotic relationship in which the host cell benefited from their presence. And so the cell develops, divides, produces new daughter cells, and the mitochondria divides and produces its own daughter cells because the mitochondria has its own DNA. It has its own ribosomes. And in fact, the DNA that it's inside there, that mitochondria can actually be traced back to your mother's side of the family. So you get your mitochondria from your mother. And because that relationship is so predictable on the maternal side from generation to generation, scientists have been able to trace back a couple 100,000 years to an actual mitochondrial Eve that they believe may have been what all the other lines have descended from. This is kind of what they think may have happened. This ancestral eukaryotic cell engulfed some type of early prokaryote, which we know now as the mitochondrion. It stayed inside the host cell, liked it there. The host cell liked having it, and then the mitochondria kind of does its own business, helps convert that sugar into energy, but it divides on its own cycle and does its own kind of things inside there. And then the last structure we're going to talk about is the cytoskeleton. The cytoskeleton is responsible for doing a lot of different things. It provides cell shape. It provides anchorage point and helps facilitate movement of organelles. Helps facilitate locomotion in different types of unicellular organisms, including amoeboid movement and movement using cilia and flagella. It also helps facilitate muscle contraction in tissues, like, for example, in us. Three primary groups of cytoskeleton structures are microtubules, microfilaments, and then intermediate filaments. Here's an example of some cilia close up. They kind of beat together little tiny hairs, which help project the protus forward, for example, forward. You also have cilia in your throat, which helps get rid of, like, dead and breaking down skin and things like that, mucus. Here's an example of a flagella, which is kind of like this long tail structure here. And the last thing we're going to talk about in the chapter are cell walls, which we find in plant cells and also in fungi. Plant cells, they are cell walls made of a material called cellulose, whereas fungi, it's made of a material called chitin. But both provide support for the cell. They also make it so the cells don't move very effectively. That's why they're kind of rigid there. But they do provide support as well. It's not the same as the cell membrane. That's a different structure, which we'll talk about in the next chapter. And in summary, here's a nice little chart here for you that's got all the different organelles that we looked at, broken down by their functional category. And don't forget to look this chart over here. Make sure that you're able to do these things for the upcoming test. Can you answer these questions? And then if you can, you'll know you'll be ready. And remember, as you get to the back part here, cross off these last couple as we ended up talking about this here. And so that concludes our chapter. And our next chapter, we'll be looking at the plasma membrane in more detail in chapter 5.